Thank you for the invitation. So I will be talking about speciation in Ficedula flycatchers, and I will focus on how we can use genomic tools to link different aspects of speciation together. Okay, so speciation is defined as a split of one population into two new populations that can evolve independently from each other. And when we are working on speciation in animals, it's important that reproductive isolation evolves, because otherwise they can't evolve independently. So when we study speciation, we can say that we study the evolution of reproductive isolation. And this can be studied at different points of the speciation process. So in the beginning, ecological divergence is very important, as we have heard before. And ecological divergence can itself lead to speciation. For example, when you have a host plant shift, then it can be very fast. So you have reproductive isolation evolving directly. Or it can interact with sexual selection, and you get assortative mating through additional processes. When we look ahead in time, quite a long time ahead, you have genetic incompatibilities evolving. And because of practical reasons, these type of processes have been studied in different types of systems. So ecological speciation has been studied in natural populations, while genetic incompatibilities has mainly been studied in the lab. But in order to find out the links between these different steps in the speciation process, it would be good to study everything in the same system. And you can also use genomic tools to link different stages of the speciation process. So we could ask ourselves, what are the relationships between the different stages of the speciation process? So how is ecological divergence related to the buildup of genetic incompatibilities between species, for example? And we can translate that into a genomic perspective and ask ourselves, so are the genes that are underlying the important traits that give rise to differences in issues, for example, is this the same genes that are later on giving rise to genetic incompatibilities between the divergence populations? So by finding out the identity of the genes, we can get an idea about how these processes are linked. And we can also ask ourselves where in the genome these genes are found, and that can give us a hint of how reproductive isolation can continue to evolve even if you have it happening in symmetry or at secondary contact, where you have some gene flows going on between the divergent populations. Okay, so this is the study system I'm using. You have the collared flycatcher, which has a more southern distribution, breeding distribution in Europe, as you can see in yellow. And then you have the pied flycatcher with more northern distribution. And they meet in two different contact zones. So we have a broad hybrid zone in Central Europe, and we have one on Öland and Gotland in Sweden, where we studied them. And as you can see on the picture of Öland and Gotland, the red dots are different nest box areas where we provide the birds with breeding nest boxes, which they do prefer when they can find them. And they both like to breed in this type of deciduous, rich deciduous woodlands. And this is just to show you how it can look in the spring. You have a nest box with a number on it. So these birds, they migrate to Africa, and when they return in the spring, the males return before the females, and they first fight over the suitable territories. And once they have established a territory, they start to sing and they attract a female. And it's only the female who's incubating the eggs, but both parents help when they feed the nestlings. And it's quite quick. So the chick in the hand is only 3 days old, and 5 days later it has grown to that size, as you can see on the next picture. And in total, it only takes 2 weeks for them to leave the nest from they have hatched. So it's quite quick. Okay, so we ring mark all the adult individuals and all the chicks. And because these birds tend to return to the same breeding site year after year after year, we can build up very nice pedigrees for genetic studies. And we also get good estimates of lifetime reproductive success, so good estimates of fitness. And we measure all the morphological traits. So for example, the white wing patch, the white forehead patch, tarsus length, wing length, everything you can measure. And we ring mark the chicks and weigh them before they leave the nest. And the way how much they weigh when they leave the nest is a good predictor of whether they are going to survive or not. Okay, so how can we use this system to study speciation? So if we start with the first component, like separation due to ecological adaptations, the idea behind that is that you have two different parental species that evolve different adaptations to the environment. So you have different combinations of traits that give rise to have high fitness. But when they hybridize, the hybrids have an intermediate phenotype, and therefore they sort of fall between two shares. So they can't utilize either parental habitat in an efficient way. And these are examples you are familiar with. So normally when people study these kind of problems that hybrids can encounter, they look at morphological traits that are associated to the type of food that the organism are using. So you have the example of the Galapagos finches, their beak sizes differ, and the hybrids have problems because they have a beak size that are intermediate and is not adaptive to either seed size. And you have the mouth structures in the cichlids. When it comes to the flycatchers, these two species are very similar in size and in shape, and they use the same foraging techniques, so they catch flying insects. But nevertheless, we thought they might have different food preferences. So we put little infrared cameras within the nest boxes, and we recorded the type of prey that the parents were bringing to their offspring. And as you can see from this analysis, a principal component analysis of the type of prey that parents are bringing, that is almost a complete overlap. So they use the same type of food. But there are some other differences, both in behavioral traits and in life history traits. So collared flycatchers are more aggressive. So when the males arrive and they compete over nest boxes, collared flycatchers tend to chase away pied flycatchers because they're more aggressive. Also, when we compare the nestlings, so we have done swapping experiments where we exchange half the brood between the two species. Because they bring such a similar food to their nestlings, we could do that. And then collared flycatchers were found to beg more intensively for food than pied flycatchers. And collared flycatchers also have smaller brood sizes. And collared flycatchers are less tolerant to decline in food. So when we look across the breeding season, the environment gets worse as you wait, because then if you breed too late, you have missed the peak in food availability. And this is very bad for collared flycatchers. So they have a steep decline in the growth rate of their offspring and also in the survival. We don't find the same decline in pied flycatchers. So these differences in life history makes them able to deal with different types of environments. And also if you look at the hybrids, they fall in between. So if you compare the performance of hybrid chicks in relation to purebred offspring, they fall exactly in between, as you would expect if ecological divergence is important. And these differences also lead to habitat segregation over time between these two different species. Because collared flycatchers are more aggressive, they push pied flycatchers away, but because they have certain woodlots that are lower quality, they can remain there because their offspring are doing better in this type of habitat as compared to collared flycatchers. So even if they would prefer the same habitat, they can still sort of cope with the poorer conditions in more pine-dominated areas. Okay, so if we move on to the next sort of component of evolution of sexual isolation, that is through sexual selection on different types of traits. And sexual selection by itself could be important for the buildup of reproductive isolation or in combination with ecological divergence. So we can use the flycatcher system because they are still hybridizing, so we can ask ourselves what type of traits needs to change in order to have complete sexual isolation evolving between these two species. And I can also mention that the male in between the two different species is a hybrid, so they have this typical intermediate plumage with a broken necklace, so they are easy to identify in the field. Okay, so what about plumage? We have shown that collared flycatchers that are more similar to pied flycatchers in their plumage, that is mainly young collared flycatchers, are more prone to hybridize than the older males that look more different to the other species. And because this could be due to other aspects like how able they are to take up territories, we also did cage experiments to really tease apart these different possibilities to show that it's actually the plumage in itself that females are looking at. So a pied flycatcher female that is sort of forced to hybridize, that can't find a mate of her own species, would prefer a male of the other species that at least look more similar to her own species in the plumage traits. There is also character displacement between the two different species, so in areas where they co-occur, the males are more different in plumage. So pied flycatchers become more brown when they co-occur with collared flycatchers. And collared flycatchers have larger forehead patches and larger white patches on their wings. And Glenn Peter Satcher showed in the past that this helps species recognition. So there is a corresponding change in female preference. So in the older hybrid zone in Central Europe, females would prefer the males that look more different from the other species. And that helps species recognition. The song is also important, but this is more complicated. In allopatry, they sing very differently. So you can see on the sonogram at the top, you have a typical collared flycatcher song, and in the bottom, there is this typical pied flycatcher song. But when they co-occur, pied flycatchers learn components of the other species' song, and they incorporate it in their own song. So they sing a sort of mixture between the two different species. So there is convergence in song, while it's divergence in plumage. And this is a very bad idea, because it leads to hybridization. So it's very common among the males that do this. So in Öland, 60% of the males, the pied flycatcher males, incorporate components of the collared flycatcher song. And in all the cases where I've recorded the song, the only cases we have found where males hybridize is when they sing this mixed song. So it's really a bad idea to do that. And it also shows that song is an important species recognition trait. Okay, let's move on to the third component, that is genetic incompatibility between the two different species. So the most popular idea about the evolution of genetic incompatibility is this Dobzhansky-Muller model, where you have genetic incompatibilities evolving in allopatry. So you have different mutations going to fixation when the two populations are in contact. And because these new genes haven't evolved together, they are not sort of functioning well together because they lack a history together, evolutionary histories together. There are also some other patterns if you look across species. So one of them is Haldane's rule, which is that hybrids of the heterozygote sex suffer first during evolution of genetic incompatibilities. And in mammals, that would be male hybrids, and in birds, it's female hybrids. So birds have this other sex determination system, and we call their sex chromosome ZW instead. Okay, so I've already mentioned this. So the flycatchers, they follow holding through. So female hybrids are totally sterile. They would behave as normal females. They pair up and they lay eggs, but their eggs never hatch. In all cases, totally infertile. Males vary a bit in their fertility. And first we thought that they didn't have any problems at all, but when we looked at paternity in their broods, we found out that they have a high reduction, so they lose paternity. And they also have problems when it comes to pairing, so females seem to find them a bit ugly, so they pair to less extent than the other males. And these problems remain if we look at further generations. So this is a sort of cumulative estimate of hybridization, of the cost of hybridization. So if you only look at the first generation, there is a reduction of 40% if you compare hybridizing pairs to pairs that don't hybridize. But if you continue and estimate fitness based on the grandchildren produce and the great-grandchildren produce, the cost is getting higher and higher. So if you go several generations ahead, they only have 2% of the fitness as non-hybridizing pairs. So there's not very much gene flow going on between the two different species. Even if the rate of hybridization could be quite high. So in certain areas we have 7% hybridization going on. Okay, to summarize so far, we have knowledge about all these three different components of reproductive isolation in the flycatcher system. We have different adaptations that lead to selection on the level of the chicks. And then we have sexual isolation, plumage traits are important, and song is important. And we also know about genetic incompatibilities. So what about the relationship between all these processes? So then we can get back to the questions I asked in the beginning. So what's the identity and function of the genes that are involved? And where in the genome can we find the relevant genes? So we don't know this yet. We have some ideas of where in the genome we should start looking for those genes. And sex chromosomes are predicted to be sort of hotspots for speciation genes for a number of reasons. So they contain fast-evolving genes. They contain genes that have sex-specific fitness effects. Their homozygosity leads to exposure of incompatible genes. And there are also some ideas that they could be sheltered against recombination. So genes that are sitting on them can remain associated despite gene flow. And there has been some specific models on speciation based on sex linkage of the relevant genes involved. And when we talk about relevant genes, we mean genes that are involved in both prezygotic and postzygotic isolation. So for example, species-specific ornaments and the preferences for the ornaments, and genetic incompatibility, or some other ecological adaptations that are leading to postzygotic isolation. Okay, so there are some indications that the Z chromosome, which is the sex chromosome in birds, is a hotspot for speciation genes in the flycatchers. So according to the patterns of genetic incompatibility, there are some Z-linked genes involved. And there is also some Z-linked genes involved that affect male coloration. And that was sort of known from the way back crosses look like. So what about the female preference for their own species? So we also use the fact that females are the heterogametic sex. And that means that female hybrids would inherit their only Z chromosome from their father. So if you had Z-linked mate preferences, female hybrids would prefer a mate belonging to the same species as their father. And this was exactly the pattern we found. Now you may be thinking about alternative explanation to this pattern, and an obvious one would be sexual imprinting. So females might be looking at the male who is feeding her, and then she would prefer a male with a similar phenotype. And in order to tease that possibility apart, we did some experiments. We were swapping nestlings between the nests, and we were also using the fact that there is high loss of paternity within the broods that are mixed broods. So there are some female offspring that has a genetic father belonging to one species and her sort of stepfather belonging to another species due to this extra pair copulations. And you could use that to tease apart these different explanations. And we did find that it was actually the genetic father that mattered. Okay, so what about the function of these genes? So we haven't identified the actual genes yet, but we have some candidate genes. So by comparing genetic variation at 73 coding loci along the Z chromosome of the two different species, we were able to identify 6 different genes that had some deviations. And 2 of them are known to be melanin, involved in melanin production, so these would be candidate genes. Okay, so this is what we know so far, so I will hope to be telling you more in the future about the possible links between the traits. We haven't reached that stage yet. But we do know quite a lot about speciation in the Ficedula flycatchers so far anyway. We know that ecological divergence is important, but not in terms of traits used to feed. It's rather life history traits that are important in this system. There is also sexual isolation driven by plumage differences and differences in song. There is genetic incompatibility according to Haldane's rule. And we have established the Z chromosome as an important genomic region that we can home in on for future analysis. And the next step would be to actually identify the genes and be able to ask these kind of questions where it's the same genes that are involved in the traits that are causing ecological differences between the species that is later on causing these genetic incompatibilities. So thank you for listening.
To this, but is there the equivalent of the lion effect in birds, in female birds or male birds, which is the homogametic sex? Is one of the Z chromosomes silenced? Is one of the Z chromosomes silenced? Or is it?
No, I don't know, but I don't think so.
Yeah.
I think they differ from mammals in that way.
Thank you.
Okay, next one is Chastity.
Thank you for a very nice talk. Do you know the background of the differences? I mean, the historical biography of the two species, did they evolve largely and a pattern?
Yeah, there have been some studies done by Glenn Peter Satter's group that sort of indicate that they have been isolating during the glaciations, but when they have recolonized Europe, they have done that from different pathways. And also there seems to be some historical events of hybridization going on between the glaciation.
I have a question about the two contact zones. If you see any differences in the character displacement, for instance, if the colors say change in different parts of the body, and do you see any differences in hybrid F1 male sterility between those? Because you kind of have two replicates of secondary contact. Is that so?
Yeah, so there are several differences. So this habitat segregation has gone further in the Central Europe zone. So the pies have been squeezed up at the top of mountains, sort of. And they're also browner in this secondary contact zone. And this weird thing of pied flycatchers learning to sing similar to collared flycatchers is also more rare. So.
Okay, so you showed very nicely how life history traits differ. But you also said that there is a segregation in habitats, and there is a difference between the efficiency of using the same diet. And this is that sort of niche segregation which is needed for having two species. No?
Yeah. So I think they have already started this by being adaptive to different climate, but they would like to stay in the same rich forest, but when they can't, pies would be squeezed out, and then they will probably become even more adaptive to that.
Okay, are there any other questions? Yeah.
I found it fascinating when you showed us the difference in the sensitivity of the two species to laying eggs, to beginning to lay eggs and breed at different times during the breeding season. That speaks very strongly to what Richard Levin's called diversity feeding on the variance in the system. And it suggests to me that that's the key to understanding the ecological difference. One way to explore that, perhaps you know the answer, would be to look at that same pattern from the perspective of non-sympatric situations, so that the one that seems to be less sensitive would become more sensitive out of symmetry, and the one that's more sensitive would become less sensitive when it was out of symmetry. And that would be the principal difference between them that would then evolve to produce diversity and stability of
Yeah.
diversity.
That's a very good point. Yeah, I agree with you. We haven't looked at it.
It's not done yet?
It's not done yet. So thank you for that.
We need a grant. We need a grant.
Okay, one last question by Svensson.
Thanks. I wonder a bit about these limited ecological differences, like feeding differences in type of food, and also habitat. Can you actually say that they speciate ecologically, or can't you exclude? I mean, can you exclude the possibility that these ecological niche differences, which are quite small, actually has very little to do with speciation per se, but they arose post-speciation?
I think if we follow the argument from before, I think they partly arose in allopatry, and I think that's important, because if you are going to have like complete speciation, I think they should actually learn to coexist. I mean, if you should have it like a permanent coexistence of both species, it's important that they have some differences to start with, and then when they come together, these differences would become larger. So I think it's something that started in allopatry and that is continuing in sympatry.
Potentially it's a matter of agreement. There was